Good morning, everyone. Welcome to the 31st edition of Soup and Science. My name is Preeti and I'm one of the organizers for this event. Before we begin, we acknowledge that McGill University is on land which long served as a site of meeting and exchange amongst Indigenous peoples, including the Anishinaabe and the Haudenosaunee nations. We acknowledge and thank the diverse Indigenous people whose footsteps have marked this territory on which peoples of the world now gather. I'm going to turn the mic over to our host, Professor John Stix, who is the Associate Dean of Research, to introduce our speakers for today.
Thank you very much, Preeti. And it's really great to see everybody here today for a very exciting session, I am sure. So we have 4 speakers today. Professor Rowan Barrett, who is in the Department of Biology and the Redpath Museum. Mr. Kusha Sareen, who is in the Department of Physiology and also the Department of Mathematics and Statistics. Professor Elliot Paquette, who is in the Department of Mathematics and Statistics also. And finally, Professor Jelena Ristic, who is in the Department of Psychology. So the way it's going to work today is that the speakers are going to give their presentations, boom, 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 1, 2, 3, 4. And after they finish their presentations, we'll go into a series of breakout rooms for the question and answer part of the Soup and Science. And Preeti will explain some details later, but I think you can, if you want to move from one breakout room to another, that should be no problem. So hold your questions until the breakout rooms, and then of course fire away. So it's my pleasure to introduce Rowan Barrett. He is an expert in conservation, ecology, evolution, and behavior. And we're pleased to have him today at Soup and Science. So take it away, Rowan.
Thank you. So it's a real pleasure to be back in and part of Soup and Science, albeit sadly without the soup. I can't. Oh, there we go. Now my slides are advancing. So so I'm a professor in the Redpath Museum and the Department of Biology, and my research program focuses on understanding the genomic basis of adaptation to changing environments. There we go. Yeah, sorry, there's a little bit of a delay with the slides. So why am I interested in the process of adaptation? Well, the biological world presents us with an incredible variety of organisms. And much of this diversity is due to the unique adaptations that make these organisms very well suited to their particular environments. Understanding how these adaptations have evolved and been maintained has been a long-standing and fundamental pursuit of ecology, evolution, and biodiversity research. So I study biodiversity, and my research team is also a diverse group of people studying a diverse group of organisms. So these are the current members of my lab, along with where they're from and what kind of organism they work on. And many of these study organisms are found in different parts of the world that have quite different biotic and abiotic conditions that bring unique environmental challenges for those organisms. And this allows us to compare between systems and gain more generality across different ecological and evolutionary contexts. And we're really interested in the process of adaptation, not just because of its role in generating biodiversity, but also because this process has really important practical applications at a time when climate change is rapidly reducing the fit between organisms and their environments. So accumulating examples such as coral reef bleaching suggest that these changes are often exceeding the physiological tolerances of populations, which means that without further adaptation, they may be at high risk of extirpation. So a lot of our work focuses on trying to understand how, why, and when populations either succeed or fail to match a particular environmental challenge. And although the survival of a population affected by environmental change will be decided by the phenotypic traits of the population, so for instance, what temperature they can tolerate, the evolutionary response in future generations is ultimately determined by the underlying genetic variation that controls those traits and is passed from one generation to the next. And this means that in order to be able to predict evolutionary responses, we need to know how genetic variation influences specific phenotypes and the functional mechanisms by which those phenotypes affect survival and reproductive success. And in my lab, we combine a variety of approaches to try to do this, spanning from genome, transcriptome, and methylome sequencing through to experiments on natural populations in the field. And I guess the punchline of all of this is that we try to take genomics out of the lab and apply it to natural populations. And the best part of this is, of course, getting to do our field work and observe our study organisms in nature. Unfortunately, that's a little difficult to do during a global pandemic, but I'm hopeful that we'll be getting our boots muddy again soon. Okay, so if you're interested in learning more about our research, you can check out our lab webpage at the link here. Thanks.
Great, thank you very much, Rowan. And I like your point about getting your boots muddy. I am ready to get my boots muddy. Okay, great. So let's move to the second speaker, Kusha Sareen. So Kusha, I did a little hunting around on the internet to find out what he was into. He's our student today, and he is interested in quantitative methods in biology. So over to you, Kusha.
Hello. Hi, my name is Kusha, and today I'm going to talk to you about calcium dynamics and myelination. Okay, here we are. Hi. Okay. So I'm going to talk to you about calcium dynamics and myelination. Last March, a lot of us found ourselves with more time on our hands than we're used to, and different people I've spoken to decided to use this time in different ways. My parents tried to learn a new language. I have friends who learn new instruments, and I tried to learn how to ride a skateboard. But have you ever wondered, what are the changes that occur in your brain when you learn a new skill like this? At the cellular level, what actually looks different? The particular research question I'm interested in answering is, how does the activity of a neuron impact its signal conduction? So neurons are enwrapped by these branching structures called oligodendrocytes that deposit a fatty substance called myelin on their surface. Myelin works a lot like rubber around a copper wire, and it helps signals pass quickly and efficiently. So the typical understanding of plasticity in the brain is very synapse-centric, but there's evidence that shows that myelin is continually remodeled even in the adult brain. For example, studies in mice show that during a new social or learning task, the gross myelin volume in their brain actually increases. So to understand this, we look to calcium. Calcium is an important signaling molecule implicated in a number of processes, from neurotransmitter release to muscle contraction. So some seminal experimental work shows that calcium transients, or short-term calcium elevations in oligodendrocytes, are actually faster when the neurons that they're enwrapping are more active. This creates a framework for activity-dependent myelin plasticity. So this is a feedback system where if a neuron wants to become more active, it starts firing more quickly. That leads to faster calcium in the enwrapping oligodendrocyte, which leads to myelin sheath growth, and then the neuron can become even more active if it wants to. To understand this, we rely on mathematical modeling. The idea of modeling is to take a mechanism and data from a system that we don't fully understand, to make a guess, and use that guess to construct a theoretical model, run a simulation, and test the theory. So the data I'm using is from a culture of oligodendrocytes, and we can identify regions of interest where we can look at the calcium activity in time. When you're faced with a dynamical problem like this, we generally make use of differential equations. That's because it's really hard for me to tell you exactly how much calcium there is inside the cell at a given point, but it's easy for me to tell you how that calcium changes because I know the receptors and the forces that govern its flux, and I know the compartments in which it exists. So for example, in oligodendrocytes, calcium in the cytosol depends on flux through the plasma membrane, and calcium stores in the endoplasmic reticulum. A simulation we might do might look something like this. So this is a branching network because we think that the branching structure of oligodendrocytes is particularly important. So we can run these, we can solve these equations in a given compartment, and then see how molecules flow to the neighboring compartment and see how the calcium changes there. So in any given compartment, a simulation might look something like this, where you have a short-term elevation in calcium. And then if you look at it in space and in time along any one of these branches, you can see how a spike like this might spread in both directions and throughout the branching structure. This work isn't done yet, but the idea is to replicate some of the key dynamical properties of calcium in these cells, which we've been able to do. Hopefully this helps us understand learning and plasticity in the brain better. Just want to quickly acknowledge Dr. Kadra and his lab and MICM for funding me over the summer. Thanks.
Great, Kusha. Thank you so much. That was super interesting. I really thought the space and time approach was really great. Thank you. Okay, so we'll move to our third speaker, Professor Elliot Paquette. And Elliot is interested in all aspects of probability. And so I'm looking forward to his talk today. Over to you, Elliot.
Unmute. Oh, there we go. Okay, hi. I'm Elliot Paquette. Yeah, indeed, I'm interested in lots of aspects of probability. The specific aspect that I'm going to talk about, and it's the bulk of what I'm interested in, is intersections of probabilities and eigenvalues. So I'm a mathematician. I can't help myself. I have to put one equation in a slide. This is the equation I'm going with. This is the eigenvalue equation. I just want to remind you what it is. What is it? It's a relationship between a matrix and a vector. And it says that in some sense, you can reduce the operation of a matrix on a vector down to a single number on a vector. They're extremely useful for many reasons. They have lots of applications. In some senses, they can encapsulate growth rates of complicated systems. In another sense, they could be used to measure energy levels. They can represent energy levels of physical systems. In a statistical setting, they can be the correlations, the strength of correlations between different sets of data. And somehow this is going to turn into something called random matrix theory. So this is the eigenvalue equation, and I'm going to make some of these things random. Now, there's an origin story here, and I need to tell you just a little bit of the origin story. The origin story is that there was a time, and this was the 20th century, they were trying to figure out how to model energy levels of complex nuclei. And you see, there's an equation you could do. You can write down sort of complete set of equations for the hydrogen atom because it's such a nice small atom. But what they really wanted to do, because they were trying to build nuclear bombs and all sorts of terrible things, is that they wanted to look at the energy levels of big atoms. So here we have thorium, you have uranium, and there was some sort of theory. Well, how do we predict these levels? Now, it turns out that those are energy levels of quantum systems, and you can write them as eigenvalues of some, in some sense, big matrix. The problem is the matrix is extremely complicated. And so a physicist asked a question, Eugene Wigner, a great physicist, he asked a question, what if we just made the matrix random? In other words, the thing is so complicated, uranium is so big, let's just make a random matrix. Maybe a random matrix produces energy levels that look like this. So he did a computation, and this is where probability starts entering in the picture. You make your matrix random. And then you can do a little computation. This is called the Wigner's semicircle law. If you actually plot the eigenvalues that you see of such an interesting matrix, you get these things. And if you zoom in, because there's thousands of eigenvalues here, you can look very, very fine into the detail, and you start seeing levels. And the idea was, okay, these levels, they should somehow correlate with the picture that we saw above. I have some work in this department. So this is some of my own contribution here. This is the stochastic Airy function. So if you were to look at this set of eigenvalues near this picture over here, you see some other set of eigenvalue correlations. Whoops, I can't zoom, but this is what I wanted to show. So I have some kind of contribution about the convergence here. Now, so that's kind of the origin story. Origin story is that random matrices were used to study high-dimensional physical systems. It turned out, in some sense, to be a success. You can actually take the energy levels of big items like uranium, or in this picture, erbium. So if you take the energy levels of erbium and you actually plot them and look at the spacings, you can see a distribution which is well matched by the predictions of random matrix theory. There's all sorts of other systems which seem to have the same effects, which is very interesting. So, for example, there's the Riemann zeta function, which is this very, very enigmatic, powerful mathematical object, and zeros are a subject of lots of consideration. And there seems to be some kind of connection, and it's increasingly being proved that this collection of zeros of the zeta function has also some kind of connection to this random matrix statistics. It's not all physics, though. So I just wanted to point out that there's another side of this, and that's a very popular subject these days, which is that random matrices just seem to be everywhere. You see, the original impetus of all this was, let's take a sufficiently complicated matrix of a deterministic object. So it's the matrix that describes the energy levels of a big nucleus. And what you see is that its statistics have something to do with randomness, even though there's no actual randomness there. And that seems to be a powerful idea in a lot of things. Sufficiently big matrices just tend to look like random ones. And you can use that. You can use that for lots of things. So, you can, this is the Marchenko-Pasteur law, which is a famous distribution of random eigenvalues that appear. You can find it, you can kind of find it everywhere. So there's a famous work of this guy, Bouchot, who went and showed that if you look at, say, stock data, you can see that this Marchenko-Pasteur law starts to appear. And that's an important effect. It appears in machine learning. Machine learning, you make large systems, which are in some sense carefully selected matrices, and they're very, very complicated, huge dimensional objects. And if you go and plot the eigenvalues of some of these matrices, well, you see the Marchenko-Pasteur law. Anyway, so that's a subject that you can use to do things. That's a realization you can use to use things. So, for example, you might try to use random matrix theory to tune your algorithms. You know that you have, if you're expecting your deterministic matrix to look like a random one, you should use that insight. So I have some work in that particular department, and this is joint work with some other McGillians. This is there's two Paquettes here. That's because this one is Courtney Paquette, and this is Elliot Paquette, and this is Kiwan Lee, who's a student here at McGill, and this is Fabian Pedregosa, also in the area. And we have a project right now which is on the behavior of stochastic gradient descent, one of the most popular optimization models, and using random matrix theory to try to show how it works. Probably that's a good point to end. I think that there's lots of possible future directions here, but random matrix theory seems to appear even where the matrices are not random. That's my moral. Okay, thank you.
Thank you very much, Elliot. Really interesting talk, really quite fascinating. And I thought it was interesting that you were looking at the element erbium. I actually know something about erbium, but
oh no,
very, very interesting. Yeah. Anyway, I'll save my questions for later. Okay, and we would like to now move to our fourth and final speaker, Professor Jelena Ristic of the Department of Psychology. And Jelena is interested in cognition and also in how neuroscience interfaces with cognition. So over to you, Professor Ristic.
Good morning, everyone. Yes, so my research is at the crossroads between social and cognitive neuroscience. And mostly we study attention, and in recent years we started studying this cognitive system in how it actually facilitates our social communication. So in these kinds of interactions that we see, how does our attention help us in nonverbal communication? And specifically, we're looking at these attentional gaze behaviors or looks that people make between one another and what that communicates. So what our research has found is that the traditional model and how this has been studied, such that single participants are put in front of a computer screen, does not really seem to capture the complexity of information that is going on during real interactions. So this is a typical task that cognitive scientists do in the lab to study the role of social stimuli, or how we pay attention to social stimuli. And this is me kind of modeling that. And while this gives us a lot of sort of control over, what we present to participants and how we measure what participants are doing, it takes away this key component, the interactive component, from the task. And so what we have found in our lab is that very small changes in these stimuli, such as, for example, changing gaze direction from direct to averted, or putting emotional expressions on a face, dramatically changes the data. And in fact, it changes the theories of attention in such a way that it could not be predicted. So what we started doing, and others as well, in this new field that we like to call interactive cognition, is to start and capture what happens when there's live persons in front of us. So instead of a computer screen, what happens when we're talking to, let's say, one person, or what we call a dyadic interaction. So here's me in my office, maybe, I think this was 2019, with my postdoc, Francesca Capozzi, who has been driving a lot of this work. And so what we see in dyadic and live interactions is that the stimulus becomes live. So it's not that we're just passively observing social stimuli, we're actually communicating and reciprocally evaluating what happens, and we're changing our behavior. And we're also continually evaluating who the speaker is, what they're saying. So our behavior and the patterns of looks, as you can see here in this video, changes based on the nature of conversation. It also changes based on who the person is, and so forth. Finally, then, we also started to look at whether the behaviors that we see during dyadic interactions scale up linearly. Oops, sorry, I have to go back. Scale up linearly to group interactions. So when we add one more person to that dyad, what happens? Well, it turns out behaviors are much more complex, because now there's more people, there's more conflicting social cues that we need to interpret and make some very quick decisions on which ones to follow. So this is, again, me and Francesca and our recent graduate, Effie Pereira, who is an NSERC postdoctoral fellow now at University of Waterloo. And so what this research has shown us is that people tend to classify groups, well, research does, but we do too, into small and large. So small groups like this one, between 3 to 5 members, and large groups are generally classified as groups about 10 or more members. And we tend to interpret social cues from these groups differently. The 3 to 5 or the small group, there seems to be a so-called heuristic-based social cue tracking. So even a minority, you can see me pointing here, even a minority cue here can be deemed socially informative, so that people, in the case of conflict, such as this, people would still follow this minority rather than the majority of social cues. However, in larger groups, attentional behavior attention seems to be guided by the clear majority. So we need about 10 out of 12 or 9 out of 12 clear majority, which then seems to be followed rather than the outliers in small groups. Finally, we also started to track some of the communication patterns that are occurring in small groups. And we recently operationalized and captured these 3 types of behaviors, what we've called social referencing, participation, and mutual engagement. And we have shown that these behaviors actually correspond quite well to the social structure of the group, individual function, as well as the function of individuals later on, such as the dynamics within the group seems to predict how people behave in later tasks. So we have define social referencing as the amount of time that the member is looked at by other group members. And it seems to denote the amount of attention that one gets. So this is Effie and Francesca looking at me, and we see social referencing occurring a lot in mother-infant interactions. We also see them occurring in social interactions when we're perhaps unsure and kind of checking to see what others are doing. These kinds of behaviors are well correlated with leadership styles and so on. Participation is the amount of time that an individual looks at the other group members during an interaction. So this is Francesca here showing participation. So this is in terms of inclusion, how much we are being engaged in an interaction. And then mutual engagement is one of these behaviors that we see differs between groups and dyads. This is when two people are engaging and the third one is not. And we see that in small groups, at least, mutual engagement does not seem to occur very often because it perhaps signals social exclusion. Whereas in dyadic interaction, it's a predominant form of this nonverbal communication. So what our research shows in this work is that social and cognitive systems operate very differently when we measure them in sort of these observer, spectator type traditional paradigms, dyadic and group interactions. And we've been working on developing ways to capture all of these actually, to capture these fundamental mechanisms and then to try and trace them through these systems of complexity. So thank you for your time.
Thank you so much, Jelena. Such a topical line of research given our current situation. And I have to say, I was really struck by your videos. The people were talking and interacting, of course, but watching their hand movements as well was so interesting. And of course, we don't really see that on Zoom. So I'd like to thank all four speakers for their really great talks. And I think one of the joys of Soup and Science is for all of us to see how many amazing things are going on in the faculty. It's really quite mind-boggling for me. So we have a series of talks all this week, and it's just really fascinating to see all the interesting things people are doing